Preliminary attempts to find a gene-protein relationship. The most fruitful early endeavours to find a relationship between genes and proteins examined the ways in which gene changes affect which proteins are present in the cell. At first, these studies were difficult, since no one knew anything about the proteins that were present in structures such as the eye or the wing. It soon became clear that genes with simple metabolic functions would be easier to study than genes affecting gross structures. One of the first useful examples came from a study of a hereditary disease affecting amino acid metabolism. Spontaneous mutations occur in humans affecting the ability to metabolize the amino acid phenylalanine. When individuals homozygous for the mutant trait eat food containing phenylalanine, their inability to convert the amino acid to tyrosine causes a toxic level of phenylpyruvic acid to build up in the bloodstream. Such diseases, examples of inborn errors of metabolism, suggested to English physician Archibald E. Garrod as early as 1909 that the wild-type gene is responsible for the presence of a particular enzyme, and that in a homozygous mutant, the enzyme is congenitally absent. Garrod's general hypothesis of a gene-enzyme relationship was extended in the 1930s by work on flower pigments by Haldane and Rose Scott Moncrief in England, studies on the hair pigment of the guinea pig by Wright in the United States, and research on the pigments of insect eyes by Aucune in Germany, and by Boris Efrossi and George W. Beadle, working first in France and then in California. In all cases, the evidence revealed that a particular gene affected a particular step in the formation of the respective pigment, whose absence changed, say, the colour of a fly's eyes from red to ruby. However, the lack of fundamental knowledge about the structures of the relevant enzymes ruled out deeper examination of the gene-enzyme relationship, and no assurance could be given either that most genes control the synthesis of proteins — by then it was suspected that all enzymes were proteins — or that all proteins are under gene control. As early as 1936, it became apparent to the Mendelian geneticists that future experiments of the sort successful in elucidating the basic features of Mendelian genetics were unlikely to yield productive evidence about how genes act. Instead, it would be necessary to find biological objects more suitable for chemical analysis. They were aware, moreover, that contemporary knowledge of nucleic acid and protein chemistry was completely inadequate for a fundamental chemical attack on even the most suitable biological systems. Fortunately, however, the limitations in chemistry did not deter them from learning how to do genetic experiments with chemically simple molds, bacteria, and viruses. As we shall see, the necessary chemical facts became available almost as soon as the geneticists were ready to use them,